In the realm of cell biology, cell death is no longer seen as purely destructive, but as a crucial process for healthy cells. This video explores various ways cells die and their importance in maintaining cell and tissue health, including autophagy, apoptosis, necroptosis, pharoptosis, and more. Autophagy is a process in which cells recycle damaged organelles and proteins. It involves the formation of autophagosomes, which fuse with lysosomes to degrade and recycle cellular components. Autophagy is important for cell survival and has implications for immune response, cancer, neurodegenerative diseases, and aging. Key regulators of autophagy include TRK1, Beclin-1, and ATG proteins. Apoptosis, or programmed cell death, is a controlled process that plays a crucial role in development and maintaining tissue health. It involves distinct changes in cell size, nucleus, DNA, and the formation of apoptotic bodies. Apoptosis can be initiated through external death signals or internal stressors, leading to the activation of caspases. The BCL2 family of proteins regulates the balance between pro-apoptotic and anti-apoptotic signals. Necroptosis is a controlled form of cell death that involves certain proteins, like RIP1, RIP3, and MLKL. It can be triggered by different factors, such as DNA damage or death receptor engagement. Necroptosis causes the cell membrane to rupture and release its contents, leading to inflammation and playing a role in various diseases. Pharoptosis is a type of cell death that relies on iron and the buildup of lipid peroxides. It is characterized by changes in cell structure and the destruction of mitochondria membranes. Various enzymes, iron metabolism, and redox balance play a role in regulating pharoptosis. This process is important in areas such as cancer treatment, neurodegeneration, immune responses, and ischemia-reperfusion injury. Studying the molecular mechanisms of pharoptosis has led to potential therapeutic approaches to target this type of cell death. Pyroptosis, a novel form of cell death, induces inflammation and is activated by inflammasomes. Unlike other cell death types, pyroptosis acts as a defense mechanism against intracellular pathogens. It involves the formation of large pores in the cell membrane, resulting in swelling, DNA fragmentation, and the release of pro-inflammatory cytokines. Gasdermin proteins, especially GSDMD, play a crucial role. Dysregulated pyroptosis contributes to infectious diseases, autoimmune disorders, and cancer. Mitophagy, a selective form of autophagy, targets damaged or superfluous mitochondria for degradation. This process ensures the removal of dysfunctional mitochondria, maintaining cellular energy homeostasis, and preventing the release of pro-apoptotic factors. Mitophagy is regulated by a complex interplay between various signaling pathways involving 10-induced kinase 1, PINK1, Parkin, NIP3, and NIX. Dysfunctional mitophagy has been linked to several human diseases, including neurodegenerative disorders like Parkinson's disease and Alzheimer's disease. The regulation of cell death is crucial in the context of stem cell biology as it influences self-renewal, differentiation, and the preservation of stem cell populations. Balancing cell survival and cell death is vital for maintaining tissue homeostasis, guiding proper embryonic development, and orchestrating tissue regeneration. Dysregulation of cell death pathways can lead to stem cell depletion, impaired tissue regeneration, and increased susceptibility to diseases. Understanding the mechanisms by which cell death influences stem cell fate has profound implications in regenerative medicine, aging research, and cancer therapeutics. The unfolded protein response is a cellular stress response triggered by the accumulation of misfolded or unfolded proteins in the endoplasmic reticulum, ER. The UPR aims to restore protein homeostasis by activating a series of signaling pathways that temporarily halt protein translation, enhance the folding capacity of the ER, and selectively degrade misfolded proteins. However, if the stress persists, the UPR can also steer cells toward apoptotic or autophagic cell death pathways. Dysregulation of the UPR is implicated in various diseases, including neurodegenerative disorders, diabetes, and cancer. Necroinflammation represents the interplay between necrosis and inflammation. When cells undergo necrotic death, their contents are released into the extracellular space, leading to the activation of innate immune responses. This activation can result in the release of pro-inflammatory cytokines, chemokines, and damage-associated molecular patterns, DAMPs, which further propagate inflammation and recruit immune cells to the site of injury. Necroinflammation plays a critical role in sterile tissue damage, chronic inflammation, and the pathogenesis of various diseases, including liver injury, myocardial infarction, and autoimmune disorders. Ischemia-reperfusion injury refers to the damage that occurs when blood supply is reintroduced to tissue after a period of ischemia, lack of blood flow. The restoration of blood flow, while necessary, can paradoxically lead to cellular damage through reoxygenation, excessive calcium influx, oxidative stress, and mitochondrial dysfunction. Cell death mechanisms, including apoptosis, necroptosis, and ferroptosis, play pivotal roles in the pathogenesis of ischemia-reperfusion injury. Understanding these processes provides valuable insights into developing therapeutic strategies aiming to mitigate tissue damage in conditions such as heart attacks, strokes, and organ transplantation. Traditionally, apoptosis has received considerable attention in the realm of cell death. However, emerging evidence points to the existence of non-apoptotic forms of programmed cell death, which can occur independently or in combination with apoptosis. Non-apoptotic cell death pathways, such as necroptosis, ferroptosis, and pyroptosis, have distinct molecular mechanisms and implications in various physiological and pathological contexts. Exploring these alternative pathways enhances our understanding of the intricate nature of cell death, contributing to advances in therapeutic interventions and disease management. The dysregulation of cell death mechanisms underlies the pathogenesis of numerous rare genetic diseases. Defects in various components of cell death pathways, including proteins involved in apoptosis, autophagy, necroptosis, or pyroptosis, can result in severe clinical manifestations. Examples of rare genetic diseases associated with aberrant cell death include neurodegenerative disorders like amyotrophic lateral sclerosis, ALS, lysosomal storage diseases, and primary immunodeficiencies. Deciphering the molecular and cellular basis of these diseases sheds light on the interconnections between cell death and genetic disorders, potentially leading to novel therapeutic strategies. Cell death is no longer considered harmful but instead a carefully regulated process crucial for cell balance and human well-being. Understanding complex pathways such as apoptosis, autophagy, and necroptosis is essential for investigating causes of diseases and developing effective treatments, thereby advancing patient care and future discoveries.